Hi there, thank you for joining. I'm Patrick Danaher. I'm a principal biostatistician here at NanoString, and I'm going to be talking about updating immune cell deconvolution for the spatial genomics era. In very brief, our first two panels here are showing the platform and the cancer transcriptome atlas panel, the 1800 gene RNA panel we're using in this experiment. I'll leave you to read about those. So the essence of what we're doing is in this cartoon on the left here. We've got a deconvolution algorithm, and I'll show you how this works. So you would begin with a tissue sample, and on the left branch, you would collect geomics RNA platform data from this sample. On the right branch, you would use single-cell RNA-seq or publicly available data to derive a matrix of expected expression profiles for all the cell types in your study. So this is the expected expression profile of a T cell, of a B cell, of a fibroblast. Then with those two inputs, the geomics data and this cell profile matrix, you put them into the algorithm and get out a spatially localized estimate of all the abundances of all these cell types in your study. So this is T cell abundance, B cell abundance, fibroblast abundance within each region of the tissue. All I will say about the algorithm is that deconvolution, it's pretty rich literature right now. Our major contribution is to replace the least squares penalty at the heart of most deconvolution problems with a log linear penalty, sort of akin to log normal regression, and that has quite a few benefits to it. At the bottom here, we've validated our approach. We've compared in adjacent slides from the same tumor our estimated cell type abundances versus expression of canonical protein markers, and we've found very good correlation. This appears to be working quite well, even in the challenging data posed by small regions within FFPE embedded tumors. But I really want to spend most of my time on the application of this method, just to show the rich data you can get from localized cell type abundance measurements across a tissue. So on the top here, you can see a non-small cell lung cancer tissue. We've taken a grid of 191 regions across this tissue. And then within each one of these square regions, we haven't just profiled that little square, we've actually collected data from it in two different ways. First, from just the tumor cells, or the cells that are expressing pan-cytokeratin, and then second, from everything that's left, so the stroma or the microenvironment. So we've got 191 regions, and in each region, we've got these paired measurements of tumor expression and stroma expression. And the importance of this segmented approach is shown on the right here. There are some genes which, CD45, EpCAM are very specific to each of these segments, to the microenvironment, to the stroma. And then these other genes are examples of genes that, you really can't interpret in mixed tumor and stroma unless you collect them separately. If Ki67 is high in tumor, that's a bad thing. If it's high in the microenvironment, that's probably a good thing. It's a totally different interpretation. You really can't disambiguate them unless you collect those data points separately. So the results of our deconvolution algorithm in this tumor are shown in the middle here. The little wheel there gives a color key for all the different cell types we're measuring. Just to make it simple, we can look at one cell type at a time. So looking on the right now, you can see these dense B cell enriched regions. B cells, not only do they cluster on one side of the tumor, but they're actually very condensed within just a few regions of the left side of the tumor. T cells, or CD8 T cells, in contrast, they tend to be enriched around the regions where the B cells are, but they're diffusing farther out through the tumor, almost in a gradient away from the B cells. So this is kind of a model where you might have these B cell enriched tertiary lymphoid structures sending out instructions, and the CD8 T cells are invading out from those regions. We see Tregs basically following the CD8s. You don't see high CD8 T cells without the inhibitory Tregs. And then macrophages have a very distinct pattern. They're spread in a low level throughout the tumor, and then they also have this pocket of enrichment in the lower right, kind of away from the lymphoid populations. So yeah, totally different pattern. We can also cluster our cell type data to come up with subtypes of the localized immune response. So here, if you look at this bar plot and heat map, showing the same data, you can see we've got a macrophage-enriched subtype, which is really dominated by these dark green macrophage bars. We've got a mixed subtype, which has T cells and macrophages and monocytes, a tiny bit of B cells. And then there's a heavily B cell enriched subtype, which also carries a lot of T cells along with it. And then there's a T cell dominated subtype, which very small numbers of monocytes and macrophages, very, very few B cells, and primarily CD8 and CD4 T cells. If you look below, you can see where these four localized immune subtypes cluster throughout this tissue. And there are spatial trends to them. The macrophage subtype favors the right side, the B cell subtype favors the left, but they're all intermixed with each other. It's not a gross gradient. It's a pretty fine pattern we see throughout this tissue. And then finally, we can compare, a natural question is, how does the tumor respond to the localized immune infiltrate? So this heat map here is showing a correlation analysis between the 14 cell types that we measure in the microenvironment compared to the gene expression in the adjacent tumor cells. So every point in this heat map is a correlation between expression in a tumor and the local abundance of a given cell type. And there's far too much information here to display in a poster, but we can see little clusters of genes that are positively correlated in the tumor when CD8 T cells are abundant versus when macrophages are abundant. And we could spend a whole other poster diving into these results. Anyway, thank you very much.